So, welcome to all of you in today's session. Uh, Yesterday, in my previous lecture, I discussed about the properties of enzymes. Now, today we will be discussing about nomenclature and classification of enzyme. This is also very interesting to discuss. So, you see that enzyme is basically designated with two names. You see that the first is very concise recommended name suitable for generally daily uses, and second is basically a more detailed systematic name utilized when precise identification of enzyme is basically necessary. So, now question arises, what is recommended name? Generally, you see that recommended name generally frequently used enzyme names typically features the suffix ending with ase attached to the substrate of the reaction, like glucosidase and urease. So, this describe the action performed, such as lactase, dehydrogenase and adenyl cyclase. However, what happens, some enzymes retain their original trivial names, such as trypsin, you see pepsin, which do not basically directly hint the kind of enzymatic functions. So, systematic name, you see that naming of enzyme by convention. So, enzymes are categorized into 6 major classes, each with numerous subgroups, and the systematic name assigns the suffix ase to a comprehensive description of the chemical reaction catalyzed, incorporating the names of all the substrate involved in the reaction. For example, lactate NAD oxidoreductase, you see that each enzyme is allotted a specific classification number. For instance, just I mentioned lactate NAD oxidoreductase corresponds to 1.1.1.27, while systematic names are very, you see that very precise and very informative, and they are sometimes become very difficult for its general use. Now, with the exception of few enzymes like pepsin, renin and trypsin, generally most enzyme names and end with generally suffix ase, generally which is added to the substrate name describing the type of enzyme's activity. Sometimes what happens, an enzyme may have multiple names or different names that might share the same name, leading to some sort of ambiguity. So, to address basically this issue and accommodate the increasing number of newly discovered enzymes, biochemists across the globe have adopted a standardized system for naming and classifying enzymes. So, basically this system categorizes enzymes into 6 classes with subclasses based on the type of reaction they catalyze, and each enzyme is basically designated a 4-part classification number. Just I gave one example, and a systematic name that indicates the specific reaction it catalyzes. The first enzyme commission, you see that enzyme commission is abbreviated as EC. So, it gave its report in 1961, and this enzyme commission basically devised a system of classification of enzymes that also serves as a basis for assigning code numbers to them. And the code numbers prefixed by EC, which are now generally widely used, contain 4 elements separated by points, and you see that there is a one example here mentioned, EC 2.7.1.1, and this enzyme is ATP glucose phosphotransferase. So, the first digit, this first digit basically represents the enzyme class, and the second digit, this second digit basically represents the subclass, and the third digit, this one 1 represent the subclass, and the 4th digit represents basically the number of subclass, which specifies the actual substrate of the enzyme, which distinguishes it from other enzymes catalyzing similar reactions. So just examples, one example is given here, EC 2.7.1, and you see this in this example. Actually systematic name of the enzyme catalyzing reaction, you see that has been mentioned, that ATP reacts with D-glucose to form generally ADP plus D-glucose 6-phosphate. So ATP glucose phosphotransferase, this is the enzyme involved in catalyzing this particular conversion, and basically ATP glucose phosphotransferase, and this name indicates that it facilitates the transfer of phosphoryl group from ATP to glucose. So this is very interesting to note, and its enzyme commission or EC number is basically 2.7.1.1, and basically this EC number provides very specific information about the enzyme. The first number 2, basically just I mentioned, indicates its class as a transferase. The second number, basically 7, denotes its subclass as a phosphotransferase, and the third number, that is 1, specifies that it's a phosphotransferase with a hydroxyl group as an acceptor, and the fourth number 1 indicates D-glucose as the acceptor of the phosphoryl group. However, what happens in many cases, enzymes are basically commonly referred to by its their trivial names. In this case, the trivial name for ATP glucose phosphotransferase is hexokinase. So try to recall where this enzyme catalyzes which step in a particular pathway. We have already discussed all these aspects in detail. So just as a question, I am leaving it here, where this enzyme basically catalyzes which step particular in which pathway. Now, according to the classification recommended by the Commission on Enzymes, according to the classification recommended by the Commission on Enzymes of the International Union Code of Biochemistry, which gave its report in 1961, basically enzymes are classified into 6 major classes based on the type of reaction they catalyze. So these major classes I will discuss one by one. So you see that the first enzyme class is mentioned oxidoreductase, and its enzyme commission number is 1, and these basically enzymes catalyze oxidation reduction reaction. It's very important to note here that this class of enzyme basically catalyze oxidation reduction reaction, involving particularly the transfer of electrons from one molecule to another molecule. Now come to the, you see the second class is transferases, and its enzyme commission number is 2. Basically these transferases catalyze the transfer of functional groups such as methyl, acyl or phosphoryl groups between molecules. So the third class is named as enzyme commission 3rd, and these hydrolyses basically catalyze the hydrolysis of various bonds by adding water molecules. These bonds generally include peptide bond in protein, present in protein, glycosidic bonds in carbohydrates, and ester bonds present in lipids. Then fourth class is lyases. Its enzyme commission is 4. Lyases basically catalyze the removal of groups from substrate, leaving double bonds behind, or the addition of groups to double bonds. Basically these enzymes do not require water for their catalytic activity. Then the third is isomerases. Its enzyme commission is 5. You see, and that isomerases basically catalyze the rearrangement of atoms within a molecule, leading to the formation of isomeric forms of the substrate. Then sixth category is ligases, and basically ligases bears the enzyme commission number 6, and they are also known as synthetases, catalyze the joining of two molecules using energy from the hydrolysis of ATP or other nucleoside triphosphate. So basically these classes of enzyme provides a systematic way of categorizing different enzymes based on their specific biochemical functions. Now I will discuss one by one with some sort of examples to get more clear picture of these different classes of enzymes. So first class, just I mentioned that oxidoreductases belongs to those enzymes that catalyze oxidation reduction reactions which involve the transfer of electrons from one molecule to another. So basically these enzymes uh, catalyze oxidation reduction reactions, and trivial names of oxidoreductases include oxidases and dehydrogenases basically. And oxidases basically utilize molecular oxygen as a hydrogen or electron acceptor, while dehydrogenases oxidize substrate by transferring hydrogen to acceptor like NAD, NADP or flavin enzymes. So additional oxidoreductases encompass peroxidases, hydrolases, oxygenases and reductases, and each enzyme playing a specific role in electron transfer reaction. So in this example, that lactate is converted into pyruvate. So basically the enzyme lactate dehydrogenase catalyze the reversible conversion of pyruvate to lactate while utilizing NADH, you see, NADH. It catalyzes the transfer of two hydrogen from the lactate. You see these hydrogen are mentioned here, and then these hydrogens transferred hydrogen are accepted by NAD and which is reduced to NADH or NADS2. So oxidoreductases basically plays a very crucial role in both aerobic and anaerobic metabolism, and they catalyze the reactions in glycolysis, TCA cycle, oxidative phosphorylation, and in the metabolism of amino acids. Now second category of enzymes is transferases, and transferases you see that are category of enzyme responsible for catalyzing transfer of particular functional group such as methyl glycoside from one molecule to another. For example, transaminases facilitate the movement of amino group from an amino acid to alpha keto acids. You see that that is shown as an example. And these enzymes play basically very essential roles in number of biochemical pathways across various biological processes. So for example, you see that hexokinase or glucokinase catalyzes the transfer of terminal phosphate group or to carbon to glucose molecule, which serves as acceptor, forming the two products glucose 6-phosphate and ADP here in this reaction. So basically this is the first step of glycolysis. Then another enzyme is alanine transaminase, an amino transferase catalyzes the transfer of amino group, that is NH2, from an alanine to alpha ketoglutarate to form basically pyruvate and glutamate. So alpha ketoglutarate basically is converted to glutamate while alanine is converted to pyruvate. So basically this alanine amino transaminase catalyze a very crucial reaction. Now next class is hydrolases. These enzymes basically catalyze hydrolysis, breaking chemical bonds to split large molecules into smaller ones. And common examples of include esterases, proteases, glycosidases, nucleosidases and lipases. So basically, you see that these group of enzymes play vital roles in degradative process within the plant cells, such as lipid breakdown by lipases and protein conversion to amino acids by proteases during the process of digestion. Hydrolysis generally break down the large molecules into fragments used for synthesis, waste excretion or energy production from carbon sources. These reactions basically convert bipolymers into monomers with some hydrolysis releasing energy during the course of reaction. Example of this category is just I mentioned here, pyrophosphatases basically catalyzes the cleavage of high energy phosphoanhydride bond of pyrophosphate in the presence of water molecule forming two inorganic phosphates. This process is essentially irreversible with the transfer of hydroxyl from HOH to inorganic phosphate. So, this is a very crucial enzymatic reaction catalyzed by pyrophosphatases. Now, lyases, this is a group of enzyme catalyzes the breaking of different chemical bonds such as carbon-carbon, carbon-oxygen and carbon-nitrogen through an elimination reaction that is distinct from hydrolysis and oxidation process. In this type of basically reaction, bonds are basically cleaved via elimination, leading to the formation of a double bond or a new ring in the resulting products. Additionally, the reverse reaction known as Michael addition is also possible. For example, lyases, just I mentioned that they break very specific bonds and through the process of elimination resulting in the formation of double bond or a sort of development of new ring. Basically, these enzymes you see that enzyme is dopa decarboxylase which convert dopa into dopamine. So, basically this dopa decarboxylase catalyzes the removal of carboxyl group from dopa to synthesize dopamine and carbon dioxide. So, this is just a very interesting example of this category. Now, isomerases, these groups of enzyme, this class of enzyme basically transform a molecule from one isomer to another isomer, resulting in the same molecular formula but with a different structural arrangement. You see that isomers could be categorized as structural or stereoisomers, and isomerases basically facilitate this conversion either through intramolecular rearrangements involving bond breakage and formation or by catalyzing conformational change. So, in these reactions basically there is a typically one substrate yielding one product with the product having the same molecular formula but differing in bond connectivity or spatial arrangement of the molecules. Isomerases basically play a very essential role in various biochemical pathways such as glycolysis and carbohydrate metabolism, and they are assigned enzyme commission number beginning with EC5. Various type of isomerizations including racemization, cis trans isomerization and enolization are the examples of the category of isomerases. And basically this isomerases also includes triose phosphate isomerase and bisphosphoglycerate mutase. And basically isomerases can facilitate the preparation of molecules for subsequent reaction like oxidation reduction reactions. They can also contribute to phosphorylation reaction pathway in Krebs cycle by preparing molecules for oxidation states. Isomerases basically enable changes in position without altering the overall chemical composition of the substrate or product. For example, one example, very interesting example is phosphoglucoisomerase catalyzes the shift of a double bond from carbon 1, you see, in glucose 6 phosphate to carbon 2 here in fructose 6 phosphate. So basically it's a sort of interconversion of an aldose and ketose sugar. Another example is here you see that phosphoglycerate mutase, which catalyzes basically the transfer of phosphate group from the carbon 3 in 2 phosphoglycerate to carbon 2 in 2 phosphoglycerate. Now ligases, as I discussed, these are also known as synthetases and catalyzes reactions by forming bond to connect, or you can say ligate smaller molecules, thereby creating larger molecules. These enzymes typically increase the energy of a system, but the hydrolysis of ATP is generally often coupled with these reactions to drive them spontaneously. Like other enzymes, these ligases basically functions by reducing the overall activation energy, often by bridging the reactions group to into close proximity, and generally examples of this class of enzyme is glutamine synthetase, catalyzes the condensation of glutamate to glutamine, and you see that the presence of ATP to form glutamine and results in also the final product is glutamine, ADP and phosphate. Another example is pyruvate decarboxylase. Basically you see that this catalyzes the condensation of pyruvate and carbon dioxide to form oxaloacetate in the presence of ATP. So this is the first step in the gluconeogenesis, the synthesis of glucose from non-carbohydrate substrates. So this is a very crucial enzyme catalyzing different reactions. Now classification based on substrate type is also very interesting way of classifying the enzymes. So you just you recall one enzyme category is proteases, also known as peptidases or proteinases. These group of enzymes that catalyze the hydrolysis of peptide bonds in protein, leading to the breakdown of protein into smaller peptides or amino acids. So these group of proteases play crucial roles in various biological processes including digestion, protein catabolism, cell signaling and other processes. And basically these proteases are classified into different groups based on their catalytic residues such as serine, cysteine or threonine, etc. And they are found in all forms of life from prokaryotes to eukaryotes. And basically plant proteases play vital roles in numerous biological processes including germination, remobilization of nutrients and defense against the pathogens against like herbivores, and some of the interesting example include cysteine proteases, and these some of the legumes also possess these proteases and they are particularly active during the germination of seeds and particularly help to degradation of gluten in cereals like wheat, oats and rye. Now lipases are a class of enzyme that catalyze the hydrolysis of basically fats, breaking down triglycerides into monoglycerides and fatty acids, and they are produced in the basically in pancreas, mouth and stomach, and they are involved in digestion, transport and processing of various dietary lipids in different organisms. Actually these lipases are group of enzymes also play very important role in different biochemical reactions. Now, amylases are the enzymes that catalyzes the hydrolysis of starch into sugar, and they are present also in the saliva of humans and some other mammals, where they begin the chemical process of digestion. But particularly, for example, salivary amylase. So there are three main types of amylases: alpha amylases, beta amylases, and gamma amylases. Alpha amylase is present in humans, animals, and plant and microbes, whereas beta amylase is uh, basically found in microbes and plants. Gamma amylase can be localized in both animals and plant. So these enzymes are used in different industries, various processing industry, particularly food processing industry like textile industry, paper, detergent, and various pharmaceutical industries. They utilize this group of enzymes, particularly amylases. Now, nucleases are the enzymes you see that they cleave the phosphodiester bonds of the nucleic acid, including both DNA and RNA. Here, they can be endo or exo DNAs, RNAs, topoisomerases, recombinases, ribozymes, or RNA splicing enzymes. One example of nuclease is S1 nuclease, that is mentioned here in the slide. You see that this group, they are involved in variety of biological processes, including DNA replication, recombination, repair of DNA, RNA maturation, processing, and various defense responses uh, inside the cell. And basically there are two primary classification of nucleases based on the locus of the activity: exonucleases, which digest basically nucleic acid from the ends, and endonucleases, which act on regions in the middle of the target molecules. So nucleases are basically both found in animals and plants and are very essential machinery for many aspects of DNA repair. Basically, restriction enzymes, you might have gone through it, are a type of nuclease that split only those DNA molecules in which they recognize a particular sequence. And this group of enzymes play a very fundamental role in the field of recombinant DNA technology and also in genetic engineering. Now, classification based on regulation, you see that and a very interesting example of constitutive enzyme is cyclooxygenase 1, named abbreviated as COX-1, which is continuously produced at a very consistent rate within the cells, irrespective of the metabolic state of the cell or organisms. Actually, COX-1, that is cyclooxygenase 1, contrast with another member of the same family, cyclooxygenase 2, which is induced by certain signals such as cytokines, glutamate, various growth factors, and other mediators. So, then inducible enzymes is, for example, beta galactosidase, present in Escherichia coli, which is produced when cells are exposed to lactose. Under normal conditions, what happens? The expression of beta galactosidase is generally low, since lactose is not typically abundant in the environment. However, when lactose becomes available, it serves as an inducer, causing the synthesis of beta galactosidase, which then breaks down the lactose into simpler compounds that can be utilized by the bacteria. So basically, this mechanism demonstrates the principle of adaptive enzyme production, where enzymes are basically synthesized only when required, thereby conserving energy resource. Now, the third is allosteric regulation. The term allosteric originates from the ancient Greek words meaning other solid objects, referring to the physical separation of the allosteric site from the active site of the enzyme. Basically, allosteric regulation is a prime example of how cells basically fine tune their biochemical activities in response to various environmental stimuli. Then allosteric regulation basically refers the alteration of an enzyme's activity through the binding of effector molecules at a site other than the active site of the enzyme. So, effector molecules can be allosteric activators which increase the enzyme activity, or allosteric inhibitors which decrease the enzyme activity. So, basically allosteric regulation plays a very crucial role in controlling the rates of biochemical reactions and maintaining the homeostasis in the living organisms. For example, hexokinase, an enzyme involved in glycolysis, glucose 6-phosphate, a product of hexokinase, can act as an allosteric inhibitor, reducing the activity of enzyme when intracellular levels of glucose 6-phosphate are high. So, this ensures that cells do not waste energy producing producing excess amount of intermediates when then sufficient energy is stored inside the cell. Then feedback inhibition is a cellular control mechanism where an enzyme's activity is generally inhibited by enzyme's end product, and this process ensures that cell can regulate the production of specific enzymes and their corresponding end products based on the needs of the cell. By controlling the level of, you see that end product, feedback inhibition basically helps prevent waste, maintain sort of homeostasis, and protects against potential harm caused by excessive accumulation of substances within the cells. The primary site involved in feedback inhibition is the allosteric site, located distinctly from the enzyme's active site. And the end product binds to the allosteric site, it causes a conformational change in the structure of the enzyme, thereby reducing or stopping its activity. An example of feedback inhibition is very interesting. It includes the regulation of amino acid synthesis, where an excess of an amino acid inhibits the first enzyme in the biosynthetic pathway, thus preventing the overproduction of that particular amino acid. So, this is a very interesting example to regulate a particular end product. So, thank you very much for your kind attention. I discussed in detail the nomenclature and classification on enzymes. Thank you very much.